Welcome everybody. We are discussing the chapter Cell, the Unit of Life, Chapter 8, Class 11th Biology. We have already discussed up to part 3 of this chapter. Today we are going to discuss the part 4 of this chapter. So far we have discussed the prokaryotic cell, eukaryotic cell, and in eukaryotic cell we have already discussed cell membrane, cell wall, and endomembrane system. Today in this session we will discuss other cell organelles like mitochondria, chloroplast, and ribosome, which are not a part of endomembrane system. So before I start my discussion, let me just revise what we have. Let me give the introduction of cytoplasm. In eukaryotic cell, the cytoplasm contains the cell inclusions and cell organelles, as I have told you. So these are the cell organelles: endoplasmic reticulum, Golgi apparatus, ribosome, lysosome, mitochondria, chloroplast. Then the cytoskeleton, which includes microtubules, microfilaments, and besides this, the appendages like flagella, cilia, and the glycocalyx capsule slime layer, which are present in the prokaryote. Okay. And besides that, the micro bodies are also there that we will discuss in our next session. So, cytoplasm, as I have told you, it is the semi-fluid structure whose component is 90% water, and it is present between the nucleus and the plasma membrane. So, once again I am showing in this picture, this is nucleus and this is the plasma membrane, and in between the plasma membrane and the nucleus, the portion of the cell present is called cytoplasm, and this cytoplasm contains the different types of cell organelles and cell inclusions. So these are different cell organelles, and we have already discussed the endomembrane system, under which we have discussed endoplasmic reticulum, Golgi complex, lysosome, and vacuoles in our previous session. So today we are going to discuss mitochondria, plastids, and ribosome. So let us start our discussion with mitochondria. Mitochondria occur singly or in groups, and their shape and size varies from 0.5 mu to 0.2 mu, and the size varies from cell to cell, and it depends upon the requirement of the energy for the cell or the physiological activity of the cell. And this mitochondria is considered as semi-autonomous organelle because it has separate protein synthesizing machinery. That means the mitochondria is the organelle which contains the DNA, which can synthesize its own protein. So the activity of the mitochondria is partially regulated by the protein synthesized by its own DNA, and it is partially regulated by the DNA of the nucleus. Okay, so that's why it is called semi-autonomous organelle. It is not under the control of nucleus completely. So it is partially controlled by its own DNA and partially controlled by the nucleus. So that's why semi-autonomous organelle. And this mitochondria were first observed in striated muscles. You know striated muscles, what are striated muscles? Which are having light and dark bands. We have discussed in tissue chapter, animal tissue chapter. So it was first observed in striated muscles of insects as granules. It was observed as granules by Kolliker in 1880, and he called them sarcosome. Sarco means muscle, zoma means body. So these are the granular bodies found in the muscles. That's why it was named as sarcosomes. Now let us discuss the structure of mitochondria. The mitochondria is a double membrane bound structure. I'll show you the picture. And the two membranes are outer membrane and the inner membrane. And these two membranes divide the lumen of mitochondria into two compartments. The outer compartment and the inner compartment. The inner compartment is called matrix. Okay. The outer membrane forms the continuous boundary of the organelle, and the inner membrane is folded upon itself, or it is projected into finger-like projections called cristae. So why these cristae are present? To increase the surface area. The matrix of the mitochondria contains circular DNA. As I have told you, they have their own DNA. They also contain RNA because the RNA are produced from the DNA. And since this DNA can synthesize the protein, so ribosomes are also present in the mitochondria. Okay. So let us discuss the diagram. Whatever structure I explained, let me discuss here. So here you can see this is the cross section of mitochondria. This is double membrane bound structure. So this is the outer membrane. You can see this is outer membrane and this is inner membrane. And this outer and inner membrane divides the mitochondrial cavity or the lumen into two compartments. So here you can see one compartment between the outer membrane and inner membrane. This compartment, which is called intermembrane space. This is the labeling. Intermembrane space is the space present between the outer membrane and inner membrane. Now coming to the inner membrane, the inner membrane is not a not a continuous membrane, continuous straight like a linear membrane. So here you can see that this membrane, inner membrane, is folded upon itself to form many finger-like projections. So here many finger infoldings are there. These foldings are called cristae. Okay, here is the labeling, cristae. And what is the function of cristae? The cristae increase the surface area of inner membrane. So when any membrane is folded, then its surface area is increased. As in case of ileum, the villi are present. Villi are the finger-like projections, the foldings, which increases the surface area for absorption. Similarly, the cristae increases the surface area, but for what? This cristae, you can see that the cristae contains some kind of particles. Okay. So these particles are called OXYSOMES or F0, F1 particles. Okay. So these particles are involved in the oxidative phosphorylation and ATP production. And that's why the mitochondria is known as powerhouse of the cell. So it produces energy in the form of ATP, and where the ATP are produced, the ATP are produced by these F0, F1 particles or oxysomes which are present on the surface of inner surface of the inner mitochondrial membrane, that is particularly the cristae. So the cristae increases the surface area so that more number of these particular particles can be accommodated and more amount of energy or ATP can be produced. And you know that ATP is called the energy currency. So as I have told you that this is the matrix, the inner membrane encloses the lumen, that is called matrix, and you can see that in the matrix the DNA. Here you can see the looped DNA, the blue color coiled structure are the DNA. DNA are present, and you can see the green color spherical structures are the ribosomes. So since they have their own DNA, they can synthesize their own protein which can regulate its own function. That's why they are known as semi-autonomous organelle. Very, very important. So this diagram is very important from the examination point of view, as well as the structures present inside it and along with their function are very, very important. And also why they are called semi-autonomous organelle. That is also very important. I hope the structure of mitochondria is very much clear to you. Now let us see oxysome. What are oxysome? I have told you these oxysome are the particles which are called F0, F1 particles attached to the inner mitochondrial membrane, especially to the cristae. They are embedded in the cristae. So let us see. Suppose that this is a cristae, okay, finger-like projection or infolding of the inner mitochondrial membrane, and in this membrane the F1 particles are there. They are also called F0, F1 particle or oxysomes. Okay. So these particles, here it is the highlighted view of this particle, F1 particle. This particle has a base, then a stalk or pedicel and a head. The stalk or pedicel and head are projected out of the cristae, and this base of the F1 particle is embedded in the inner mitochondrial membrane. Okay, and they are involved in the production or formation of the ATP, adenosine triphosphate, the energy currency, which is a endergonic process. Endergonic process means the process, the reaction in which energy is absorbed. Okay. The next, the function of mitochondria. I have already told you mitochondria are referred as powerhouse of the cell because they produce 95% of ATP. And these ATP are the energy currency. And how these ATP are produced? The ATP are produced during the breakdown of food molecules. That is during glycolysis, oxidative decarboxylation and oxidative phosphorylation. So these processes I have uttered here, glycolysis, oxidative decarboxylation and oxidative phosphorylation, that we'll discuss in plant physiology and animal physiology in details. And you know that the breakdown of food and formation of energy or ATP is called respiration. So, the mitochondria is also known as site for cellular respiration. Very, very important. It is called powerhouse of the cell because it produces ATP, 95% of the ATP. And it is also called site for cellular respiration because this is the site, this is the organelle where the food molecules are broken down to produce energy, that is ATP. And the intermediate products of cell respiration, that is during this breaking down of the food or oxidation of the food, whatever products, intermediate products are obtained, they are utilized in the formation of steroids, cytochromes, chlorophyll, etc. And this is also the site for production of some of the amino acids because I have told you that they have their own DNA, so they can synthesize their own protein. The mitochondria contains electron transport system which is present in the inner mitochondrial membrane. Very, very important. The electron transport system is present in the inner mitochondrial membrane. Now the next most important organelle, which is almost similar to mitochondria, that is called in most of the aspects, that is plastids. But these plastids are present only in case of plants and algae and bacteria. So these plastids are also semi-autonomous organelles like mitochondria because they have also their own DNA, RNA ribosomes. Like mitochondria, they have double membrane envelope. And very important children, these are the largest cell organelles in the plant cell. It was discovered by Haeckel in 1865, but the term plastid was first used by Skimper in 1883. And in the plastid, there is a well-organized system of grana and stroma. I will show you the picture and explain you what is grana and what is stroma. So, first of all, the organized system of grana and stroma, that is the matrix in the plastid, was first reported by Devon Wettstein. Okay. So let us first discuss the types of plastids. The plastids are of three types: leucoplast, chromoplast and chloroplast, and this classification is made on the basis of the type of pigments present in the plastid. So the first one is leucoplast. The leucoplasts are the colorless plastids. They don't have any pigments. These are non-green cells. They are present near the nucleus and their function is to store the food materials. And these cells, these part of the cells are not exposed to the sunlight, and very important children, depending upon the type of food stored by the leucoplast is of three types. Very important children, from the examination point of view, the leucoplasts are of three types depending upon the type of food material stored by them. First, a myeloplast, which synthesizes and stores starch grain or carbohydrates. A lyoplast, it is also called lipidoplast or oleoplast, which stores lipid and oils. The third one is alluroplast or proteinoplast, which stores the protein. So these are the three types of leucoplast. So very important children, again I'm repeating, these are colorless plastids, occur near the nucleus, non-green, present in the non-green cells, and they store the food materials. They are of three types: amyloplast, elioplast and alluroplast. So here you can see these are the different types of plastid, but I will show you the leucoplast only. So here you can see this is amyloplast, which stores starch or carbohydrate, lyoplast, which stores the fat droplets, then proteinoplast, which stores the proteins. Okay. Then the next type of plastid is chromoplast. This chromo means colored. So these are the plastids which contains different colored pigments other than green. So they are known as chromoplast, and these plastids are red, orange, yellow, etc. colored due to the presence of a particular type of pigment that is known as carotenoid, and they are present in the petals and fruits. And let me tell you that leucoplasts are present in the colorless plant parts of the plant. So here you can see in this picture the chromoplast, which contains different colored pigments other than green color. Okay. Then the next most important type of plastid is chloroplast. So let me first show you the picture. So this is the chloroplast. So here are the three types of plastids. As I said, leucoplast, chromoplast, and the most important one is chloroplast. So this is the highlighted picture of chloroplast. So let us discuss the structure and function of chloroplast in details. The chloroplast was discovered by Sachs and named by Skimper. Chloroplasts are green color plastid due to presence of chlorophyll pigment that is known to all of you. And these chloroplasts occur abundantly in green leaves and green part of the shoot. And the most special feature of this chloroplast is that they trap the solar energy which is used for synthesis of food. So they are known as the site of photosynthesis. Very, very important. The chloroplasts are of different shape. They may be cup-shaped as in case of Chlamydomonas. They may be stellate-shaped as in case of Zygonema. They may be collar or girdle-shaped as in case of Phyllothrax. They may be spiral or ribbon-shaped as in case of Spirogyra. They may be reticulate or net-like in case of Odogonium. They may be discoid in Boucheria. Then they may be spherical as in case of Chlorella. They may be biconvex or ovoid in case of Angiosperm. So here you can see different shaped chloroplast. This is Chlamydomonas, which is having cup-shaped chloroplast. Here you can see discoid shaped chloroplast. It is restricted to the peripheral part of the body. Here you can see this is C-shaped chloroplast. Then here you can see the stellate-shaped, star-shaped chloroplast. Then spiral-shaped chloroplast in case of Spirogyra. Then here you can see the chloroplast forms a reticular structure, network-like structure, and here it is ribbed structure in case of Phyllothrax. Okay. So these besides these shapes, there are so many other shaped chloroplasts are also there, but I have given few. Now let us discuss the structure of chloroplast in details. As I have told you, they are double membrane structure. Both the membranes are smooth. The inner membrane is less permeable than the outer, but rich in protein, especially carrier protein. The inner membrane is less permeable but rich in carrier proteins. The intermembrane space, that means the space between the outer membrane and inner membrane, is called periplastidial space. And the space that is enclosed by the inner membrane is called matrix. The matrix the portion of the matrix is divided into two parts, that is grana and stroma. What is grana? This is the inner plastidial membrane of the chloroplast, which is invaginated, means folded to form a series of parallel membranous sheets. These are called lamella, and this lamella forms a number of oval shaped closed sac called thylakoid. I will show you the picture so that it will be clear to you. Along the inner side of the thylakoid membrane, there are a number of small rounded semi-crystalline bodies called quantosome. What is quantosome? Quantosome is the photosynthetic unit, the exact site where the photosynthesis takes place. Let me show you the picture. So here you can see this is double membrane bound structure. The membranes are very smooth. From the picture it is very much clear. So this is outer membrane, this is inner membrane. In between the outer and inner membrane there is a space you can observe here, and this space is called periplastidial space. Now the inner membrane encloses a cavity or lumen, which contains the matrix. And in the matrix, you can see the lamellar structures, and these lamellar structures are invaginated and that is folded, and you can see the stacks or pile-like structures. So here you can see these are pile-like structures, and these pile-like structures are called thylakoids. And in the thylakoid membrane, in the membrane of the thylakoid, the quantosomes are present. The quantosomes are the photosynthetic unit, the exact site where the photosynthesis takes place. And the stacks of thylakoid is known as grana. So here you can see these are the stacks of thylakoids. These thylakoids are called grana, and individual coin-like structure is called thylakoid. So don't confuse children. Individual coil invaginated structure is called thylakoid. The piles of coin-like structure or thylakoid is called grana. Then the matrix part is called stroma, and in the thylakoids or the grana are connected to each other with the help of stroma lamella, which are shown here. It is stroma lamella, and then this is outer membrane and inner membrane. So this is the structure of chloroplast. And besides that, in the stroma, the DNA is present, ribosomes are present, and RNA are also present. So this is another picture of chloroplast. Here you can see that this is the stroma, the matrix is stroma, and where the grana are present, these are coin-like structures, and the stacks of coins is called grana, and individual coin-like structure is called thylakoid, and this stroma contains the lipids, starch granules. Besides that, it contains DNA, ribosomes, and since it has its own DNA, it can synthesize its own protein, so it is also known as semi-autonomous organelle. So children, remember, there are two organelles present in the cell which are semi-autonomous. What are they? They are mitochondria and chloroplast. But the chloroplast is present in the plant cell. So if it is asked in the plant cell, what are the semi-autonomous organelles? Then what will you write? You will write mitochondria and chloroplast. But if it is asked in the animal cell, then it is only mitochondria. So I hope the structure of chloroplast is clear to you. Now we'll discuss the structure of the stroma and what is the function of stroma and grana and the pigments present in the chloroplast. The stroma, as I have shown you, it is transparent. Okay, this part is stroma, and it is rich in protein and it is a watery substance. Most portion of this stroma is water. And you know that the chloroplast is the site for photosynthesis. The photosynthesis involves through two processes, two phases. One is dark reaction and other is light reaction. In the stroma, remember children, very, very important. In the stroma, the dark reaction of photosynthesis takes place, whereas in the grana, the light reaction of photosynthesis takes place. And this stroma is filled with a particular type of enzyme that is called Rubisco, that we will discuss in photosynthesis chapter in details. So I am not talking about Rubisco enzyme and its function here. So just for the time being, you just know that you should know that it is an enzyme which helps in dark reaction. Okay. So now let us see what are the pigments present in the chloroplast. You know that the chloroplast contains the green pigment, that is chlorophyll pigment. But these chlorophyll pigments are of different types: chlorophyll A, Its formula is C55H72O5N4Mg, and its special feature is that it is with a methyl group. Very important for your NEET examination. Then chlorophyll B, its formula is C55H70O6N4Mg, and it is with aldehyde group. More often in the NEET examination, it is asked what is the difference between chlorophyll A and chlorophyll B, or in which of the following chlorophyll pigment the methyl group is present or aldehyde group is present. Then you can write, excuse me, you can write that in chlorophyll A methyl group is present and in chlorophyll B aldehyde group is present. Besides that, two more types of chlorophyll pigments are present. They are chlorophyll C and chlorophyll D. Okay. Now let me show you the thylakoid and that quantosome. I was talking about the quantosomes. So here you can see these are the thylakoids, and these are the stacks of thylakoid. And in the membrane of the thylakoid, different types of pigments are there. Okay, so here you can see the chlorophyll pigments, how they are arranged in the thylakoid membrane, and that constitutes the quantosome, and that is the actual site for photosynthesis. And all these pigment systems together constitute a photosystem which absorbs the light, energy and helps in converting them to the the production of ATP. Okay, so this is the site of the thylakoid or the membrane of the thylakoid, which contains the different types of pigments, chlorophyll pigments, which together constitute the photosystem, and this photosystem absorbs the light and performs the light reaction of photosynthesis. And the details of how the light reaction takes place by the chlorophyll pigment, what is the reaction center, that we will discuss in plant physiology. Now let us discuss some of the other pigments present in the chloroplast other than chlorophyll pigment. In bacterial chloroplast, there is a specific type of chlorophyll pigment is present, which is called bacteriochlorophyll. Its formula is C55H74O6Mg. It is also called chlorobium. And these are present only in photosynthetic bacteria. Then other type of pigments present in chloroplast are carotenoid. These carotenoids are hydrocarbons soluble in organic solvent, and these carotenoids are of two types. One is carotene, which is the derivative of vitamin A, and its color is carrot color. Then other type of carotenoid is xanthophyll, which is yellowish in color, and it also it is of different types like violaxanthin and fucoxanthin. So they may be violet color or brown color. So these are the different types of pigments present in the chloroplast other than chlorophyll pigments. Now the functions of chloroplast, it is the site of photosynthesis, that is for both light reaction and dark reaction. I have already told you light reaction takes place in grana, very important, and the dark reaction takes place in the stroma. And here the breakdown of water in presence of light takes place. The reduction of NADP, nicotinamide adenosine diphosphate, takes place in the grana. The Photophosphorylation or the synthesis of ATP in presence of light also takes place in the chloroplast. They store starch and it is the factory of synthesis of sugar. The function of chloroplast we'll discuss in plant physiology in details. Then the next cell organelle is ribosome. The ribosomes are the smallest cell organelle which is without membrane. The ribosomes are composed of the components of ribosome are the protein and RNA, and they are attached to the rough endoplasmic reticulum mostly, or they may be floating freely in the cytoplasm. And they are known as the protein factory of the cell because this is the site where the protein synthesis takes place. So this is the structure of ribosome. You can see that both in prokaryote and eukaryote, the ribosome consists of two subunits. One is larger subunit, other is a smaller subunit. Again, these subunits are composed of many smaller subunits that we will discuss later on. So, this ribosome has there is a tunnel in between the two sides through which the mRNA passes during protein synthesis. And these ribosomes are attached to the surface of endoplasmic reticulum during protein synthesis in case of eukaryote. But in case of prokaryote, there is no endoplasmic reticulum. So, a number of ribosomes are held together with the help of a single mRNA to form a structure that is called polysome or polyribosome. So here you can see how it is helpful in formation of protein. Here you can see that this is the site through which the amino acid enters into the raw material for protein synthesis, enters into the ribosome, and this is the site where the peptide chain is formed and released. Okay, so this how this is involved in protein synthesis that will be discussed in class 12 in details. Now these types of ribosome, they are of mainly of two types: 70S ribosome which is found in prokaryotes, mitochondria and plastids of eukaryote. And 80S ribosome, it is found in the cytoplasm of eukaryotes. Very important children, you have to remember what is 70S and 80S ribosome. So here from the picture, let me show you that prokaryotic ribosome is 70S type, eukaryotic ribosome 80S type. What is 70S, 80S? I will just explain you. Again, this 70S ribosome consists of two subunits, that is 50S and 30S. So don't put the mathematical formula here that 50 plus 30 is 80, how it becomes 70. It is not like that. It is not a mathematical formula. It is something different I am going to discuss. Again, this 50S subunit composed of 5S RNA and 23S RNA. The 30S subunit consists of 16S RNA. Similarly, in eukaryotes, the ribosome is 80S type, consists of larger subunit 60S, smaller subunit 40S. Again, 60S consists of 5S RNA, 5.8S RNA and 28S RNA. Excuse me. The smaller subunit 40S subunit consists of 18S RNA. So this is the difference between the ribosome of prokaryote and eukaryote. Now let me tell you what exactly this S is. The S stands for sedimentation coefficient. So when the scientists wanted to identify the structure, that is ribosome, they centrifuged the homogenized cell. Homogenized means the fragmented cell. So when it is centrifuged, centrifuge is an instrument where there is a rotor at the center, and the rotor, the test tube holders are there, and in the test tube we have to take the fluid which contains the fragments of the cell. So when it is rotated at a high speed, in a faster rate, then the substances present in the fluid sediment as per their size and density. So more is the density of the particle, faster is their sedimentation. So this sedimentation coefficient can be defined as the rate of sedimentation of any particle, that is in a particular time. So here, if a substance is having a mixture of in a mixture various substances are there, and when we want to separate them by centrifugation, then the substance which is having more density will sediment faster. So their sedimentation coefficient or sedimentation rate is high. And if they are massive, then also their sedimentation rate is high. And if frictional coefficient is more, I mean the size of the particle is less, more friction is taking place, then the slower is the movement, and if more centrifugal force is there, then the faster is the sedimentation. Now let us see what exactly S stands for. S stands for Swedberg units. It is the measure of how quickly particles sediment in an ultracentrifuge. I have told you, while centrifuging, during centrifugation, the particles present in a mixture, in a solution, settle down in the test tube depending upon their density, depending upon the size of the particle. Larger the particle, greater is its S value. Very important, children. So, smaller subunit of ribosome sinks slower than the larger subunit. Okay. So, I think what is S or sedimentation coefficient or Swedberg unit is very much clear to you. So, here I will make you more clear. So, this is here we have taken blood, and you know that blood contains plasma, then the clotting proteins are there, then different types of cells, that is WBC, RBC and platelets are there. Suppose we want to separate these RBC, WBC and plasma, then we have to centrifuge them. So this is a centrifuge machine where there is a rotor, this is the rotor, and this is the test tube holders, and in the test tube we have taken the blood. Now when it will rotate at a specific revolution per minute RPM, then at different time the different cells will sediment. So here you can see the RBC has been occupied the lowermost position. That means the RBC are more in quantity, so the density of RBC is more, so they are settled down. They sediment faster. But if you compare the sedimentation rate of WBC with RBC, then the sedimentation rate of WBC is less because its density is less. Similarly, you can see that the plasma occupies the highest position because the plasma is less dense, so it occupies the upper position. So here more clearly, let me explain. So here suppose say we want to separate the different organelles or different parts of a cell. Then we have taken a tissue or cells here which is homogenized, that means fragmented. The cell is fragmented. So what we'll observe when it is centrifuged, here you can see I have shown you differential centrifugation. Differential centrifugation means we have to stop the centrifuge machine after a fixed time. So here you can see that after 10 minutes, what has been settled down? The pellet containing nuclei and the cellular debris are settled down. That means their density is more and their rate of sedimentation is high. So after 10 minutes, these substances are sedimented. Okay. Now you can separate this sediment. The next, again when you will centrifuge for another 5 minutes, that is after 15 minutes, what you will get? You will get that the pellet containing mitochondria and chloroplasts are settled down. That means the nucleus is the most dense part, then the mitochondria, then the microsomes, then the ribosomes. So in this way you can see that the sedimentation rate is highest in case of nucleus. Okay. So here 100 gram per 10 minute. In 10 minutes 1000 gram. 1000 gram has been settled down. But here in 15 minutes that is 20,000 gram. So in this way you can compare the sedimentation rate of different organelles of the cell and accordingly they are named. Okay. So here you can see the sedimentation coefficient of the substances varies from cell to cell and that depends upon the density and massiveness. So these are the chemical composition of ribosome. I have already told you in 70S. These are the composition. No need to go through such a huge data. So I have already told you. If you want, then you can pause that slide and you can note down. Now, function of ribosome. Ribosomes are also called protein factories of the cell. I have already told you. The enzyme peptidyl transferase is present in the larger subunit of ribosome, which helps in protein synthesis. So, children, in this session, we discussed three most important cell organelles which are not a part of endomembrane system. They work individually, out of which the mitochondria and plastids are the semi-autonomous organelles. Very important point to be remember. Then ribosome. In ribosome, the function of ribosome, the location of ribosome and the structure of ribosome is also important. So, in our next session, we'll discuss the cytoskeleton, then the appendages, that is cilia and flagella, centrosome and centrioles, nucleus, the control room of the cell. Then we'll discuss the microbodies. So, besides microbodies, in plant cells, the spirosomes are there. And these microbodies also includes the glyoxysome, peroxysome, that also we'll discuss. So, thank you children for watching this video till end. And wherever you have doubt, you just pause the video and listen to my discussion, my explanation, go through the diagrams, your doubt will be clarified. Even then, if you have any doubts, you can write in the comment section. In regular interval, I go through the comments, and if any doubt is asked by any students, then I definitely give reply to that comment. So children, don't forget to like this video, and please comment. Thank you once again for watching this video till end.